notices that this is being recorded. For those of you who are comfortable doing so, I'm going to try to be a little bit interactive. So if people are willing to turn on their videos, that'd be great. I totally understand that some people are in situations where that's not comfortable, and that's totally fine too. So for those who don't know me, my name is Matt Simon, and I'm a chemical biologist in the molecular biophysics and biochemistry department. And today I get to have the fun of talking with you guys about genomics. And just to give you a little bit of background, my lab does a lot of technology development, and a lot of what we do ends up being sequencing-based technology. So there are certain aspects of genomics that I know well and other aspects that I know less well, and I'll try to be clear when I'm talking as an expert. So let's get started by thinking a little bit big picture before we go further. And that's the question, this is the first set of DADU modules, and the question is, what actually is genomics? What are we, set out to learn today's lecture and the one on Monday? And let me say that you're welcome to unmute to answer my questions. If any of you are uncomfortable doing that in front of the whole class, Jai Hao has generously offered for you to chat at him, and he can read questions or responses. So what is genomics?
Study of all genes.
Sorry, can you say it one more time? I think I cut out for a moment.
When we're trying to study and look at all genes in a particular organism.
Yeah, exactly. So all genes, and what do we call it when we're looking at all genes in an organism? Genome. And I would say that's important in part because, gene has, people can debate what it is, but also we care a lot about non-coding regions and coding regions. And so the entire genome, frequently is what we're doing. So I think the definitions that I'm going to stick with for the purpose of this lecture is to say that genomics is a global study of how biological information is encoded in genome sequence, right? And so as Raghav, I think, said, that includes genes, but also, of course, the regulatory sequences, as well as all the genetic variation that's there. However, there's also some operational definitions. So I would say this is a relatively formal definition about what genomics is. If you think experimentally about what people are doing when they say they're, doing genomics, what do you tend to think of? Anyone can hop in here.
Sequencing?
Yeah, exactly. So, frequently genomics, refers to people who are looking at sequencing, and there's a reason for that, right? Because part of what we care about is how do we get the global, study of the information to work out. And certainly sequencing is not the only way. And I want to be really clear that I'm going to focus on sequencing in a lot of this lecture, but I don't mean to mislead you to think that sequencing is, the only way one can study genomes. There are many other ways, including microscopy approaches that we might have a chance to talk about on Monday a bit. But so the idea is that the way the information is read out to produce distinct biological outcomes is an important part of this. And because we care about how biology reads it out, we also care about how we as experimentalists read this out. So in practice, even though the formal definition has to do with information content and how it's read, in practice, frequently when people are discussing genomics, they're referring to the operational definition of people who are doing a lot of deep sequencing. And I think both these definitions are valid and are worth considering, but obviously they take on different mindsets. So as we're going forward here, of course, the goal of this, right, is to go, and biology is actually molecules, right, and principles and how these things come together in organisms. And yet the information content, especially the genome, is A's, T's, C's, and G's. And so, a lot of these, today's lecture and the one on Monday is going to be about the relationship between these two and, how we can learn about that relationship. So today I'm going to focus mostly on the sequencing technology itself and kind of the really kind of what I would say vanilla genomics of, thinking about how we sequence DNA. And then on Monday's lecture, we're going to talk much more broadly about ways that you can use these same tools to ask all sorts of questions about genomes or annotations or functions and other bits of biology. So the real learning objectives that I have for you guys, and as a wet lab scientist who's a chemist, is, I see my role here as helping you guys think about the information that you're going to process when you're working with genomics data. And so generally speaking, that means that, there's a bunch of samples and organisms and things like that. And at some point we take that and we turn it into information in terms of A's, T's, C's, and G's. And that's kind of going from wet lab to FASTQ, which is just a name of one of the common sequencing formats that you guys might work with. And so that's trying to understand that link is really a big part of today's lecture. But also, of course, the idea is why do we care? And so the applications of studying genomes and everything else that you can study with biology using sequencing is there. And what I hope to convince you of, if I'm successful in this set of lectures, is that a sophisticated use of the information, the genomics data that you're doing really requires not just an understanding of the data analysis, modeling, mining, all of those things, but also an understanding of the biological experiments, the sample preparations, and how those things interface with the data. So this is kind of the first data module, and I hope I will convince you that we do care about biology and chemistry, even though the focus is going to be on the data. Okay, so just to kind of motivate things a little bit here, I think all of you have had this experience, which is you all are here in part because you read some sort of academic journals and topics that you care about. And so this happens to be me flipping into the most recent online edition of Cell a couple of days ago and just going to one of the first papers that interested me. And the first paper that happened to interest me because my lab studies RNA metabolism was an interesting paper that had to do with what happens when you treat cells with drugs that disrupt splicing, and specifically how specific types of cancer cells respond to those drugs. And this isn't an important point for this lecture, but my point is I think each one of you can kind of imagine opening up your favorite journal and flipping to it. And I would bet that it would not be rare that you would have the same experience I had, which is that when you look at this based on the biology and the topic you're interested in, you end up with a journal like this and you go to figure 1A, and here people are asking what changes in the expression levels, meaning what RNAs are up and down in these particular cells when you treat them with an inhibitor. And so if you want a global answer to that, they do an RNA sequencing experiment. And we're going to talk a lot more about RNA sequencing, especially on Monday. But the point is that figure 1A is often a global characterization of some sort of perturbation that's either hypothesis directed or based on some sort of, essentially fishing expedition where people end up, making a figure like this. And that's actually the launch of a huge number of scientific studies. It's also a tool to look at mechanism. And importantly, if you're interested in this particular paper, which I am, but I imagine you may or may not be, but you probably have your own paper that has a structure similar to this, you'll also notice in these papers that pretty much all of the papers these days that involve sequencing data, which is a huge fraction of the biomedical papers out there, also have statements that say the data and code are available. And if you look at these statements, they have these codes in them. And essentially what that means is that all of you who are interested clearly because you're here in, data mining and understanding modeling of biological data, you can go in and you can take any of these studies and you can start working with the data. But in order to do that, of course, you need a sophisticated understanding of what this data actually is. And so you can go, and if you click on those links, or if you take those links and go to the databases they reference, this is actually a screenshot of the webpage that you can go to, right? And you guys can all find your favorite entry from your favorite journal, and you can go in and you can download data, and as long as you have the right toolkit, you can take it and you get files that look like this. And at first it might just look like a bunch of gobbledygook, But if you look a little bit more carefully, I think those of you who know the DNA bases won't have a hard time seeing that some of these lines have A's, T's, C's, and G's on them, and maybe some N's. And in fact, it's pretty clear that here we have a whole bunch of data that has to do with genomics. And so, what I'm saying here is that if you can understand what this is and how people collected it, you're in a position where you can immediately start doing your own independent analyses and asking your own biological questions about whatever hypothesis you want. So this is kind of to motivate how we're going to think about this is what do you want to know and what's important to know about how they collected this data in order for you to be, sophisticated users of it. And one thing that I've learned from teaching in this class kind of over time is that year to year, it seems like different students are taking this class for different reasons. So I'm going to address two questions here. One is where do all these data come from? And the other is how does the way we collect it influence what we know? But in order to answer those questions at a level that's appropriate for the class, I'm also going to start out by just doing a quick poll. And I know that you guys have a welcome survey that you're doing, but because I'm only lecturing in a couple of these, I don't know the answers to these questions. So I was going to go ahead and just launch a poll. And this is just as an anonymous poll. And the point of this is just for you guys to give me a sense of how much wet lab experience the class as a whole has with their with deep sequencing. And this just will help me kind of calibrate the way I talk about the material in the rest of this lecture. Give you just a few moments. I think and I'll have a few polls throughout this. This is just a way, it's hard to engage over Zoom. So this is one of my ways to get feedback from the class, since I can't tell whether people are nodding or not. All right. Great. I think that most people have done. So I'm going to go ahead and end the poll. It's okay if you didn't answer, and share the results. And so can everyone see the results? At least of those. Okay, I see people nodding. So what you'll see is that there's a mixture. There's a few people who have done, or a couple of people who've done at least one deep sequencing data or experiment, but there's a lot of people who have done zero molecular biology and or they've done molecular biology but only limited to non-deep sequencing. So this is really helpful to me. And I'm just going to ask one more poll that's kind of related to this, and I think I can guess the answer now. I know the results of the last one, but this has to do not with your wet lab work, but rather your analysis work, whether you've ever worked with sequencing data, regardless of whether you have any experience. Actually, I'm really glad I asked this because it's not, the answers I'm seeing already are not what I expected. So this is good. All right, cool. That's enough. Don't worry if you didn't get a chance to answer. I think you'll get the point here. Oops, you actually probably could see that right now. Anyway, so pretty much what you'll see is that it looks like actually a number of you have worked with some deep sequencing data. So that's really cool. And it means that there's a handful of people. Don't worry at all if you answered A in both of these. I will try to make sure to give a broad set of introductions to things. If you happen to be someone who answered A in both of these, please don't be shy about interrupting me and asking questions or chatting questions to Jahao so that you can get make sure that your questions are answered. But that's great. So I want to pause for a second. So if you're one of the people, one of the 7 people who says that you've been working with a lot of DNA sequencing data, or someone who has already done some deep sequencing data, do you mind just unmuting and sharing some information about what you've been doing with sequencing and what questions you've been asking?
So I used to work in studying metabolism and mitochondrial metabolism in general. And so I would be using a lot of RNA sequencing data along with and couple it with other omics data such as proteomics and metabolomics to understand how sort of cancer cells behave different from normal cells.
Great. So you were doing RNA sequencing with deep sequencing data. Do you remember what platform you were working with, like where the sequencing data came from?
This was Illumina HiSeq, if I'm not wrong. Yeah.
Great. Yeah, and in fact, as you'll see, I'm going to focus a bit on Illumina platform, but I'll also comment on some of the other deep sequencing platforms that have picked up steam in recent years. Anyone else want to tell me? I'm just kind of curious about the breadth, and it's also, I think, hopefully fun for you to learn what your classmates are doing, because I mean, it's cool. Everyone here has different arcs, and it's fun to hear about the science going on.
Yeah, I used to work with a lot of Illumina sequencing data. It was for RNA-seq, looking at kind of like RNA structure and like motif searches and things like that.
Oh, cool. So RNA structure, you mean different splice isoforms, or what do you mean by RNA structure?
No. So this was bacterial RNA data. So using like a Vienna RNA and things like that.
Oh, I see. So you
Like,
were
yeah,
actually
different
using
sorts of, like,
annotations.
yeah, secondary sequence or secondary structure. Sorry, like looking at that.
Great, good. So there's some good RNA people here, which will make Monday. Today will mostly be DNA, but on Monday we'll talk more about RNA applications. So I'm happy to hear that there's some people who care about that. Anyone else want to volunteer what they've been doing?
I did an internship where I was searching for microRNA binding sites in genomes of cancer cells, and then also searching for candidate sites for CRISPR to be able to modify those binding sites.
Oh, cool. That's really neat. Okay, so more RNA people, but in that case, you also cared about the genome since you were actually CRISPRing into the genes that encoded these things. Cool. Has anyone done any long read deep sequencing in the class, like meaning Illumina or Oxford Nanopore? No, not yet. Okay. I'll touch on that. I was kind of hoping, I've done almost exclusively Illumina sequencing in my lab, and so I was hoping there might be some experts in the room, but we'll go through and we'll talk a little bit about them because I think there's some cool directions to go. So let's go forward. But once again, I want to just remind you, I really do like when this is interactive. I just feel like it's more fun for everyone, and also it'll help keep the level of what we're talking about kind of geared towards you guys if you do engage me. So please do feel free to interrupt or to chat questions. So let's talk a little bit about sequencing experiments. So there's kind of the workflow, right, to get the data. And, I break it up into 4 steps. And, there's kind of course, isolating the sample. So where's your DNA coming from, right? And I as a chemistry PhD student, I always feel some need to remind people that sequencing, we reduce these things to information, but there's very real chemical structures that, and I like, there's one organic chemist who kind of called the Human Genome Project one of the largest natural product elucidation experiments ever done, which I kind of agree with. So the sample itself, of course, which is the real wet lab stuff. And there's a library preparation, which is also in the wet lab, of how do you take your kind of messy sample and get it into an instrument that can tell you the sequence of it. There's, of course, the sequencing itself, so how that's done. And the picture I have here is, an Illumina sequencer, but we'll talk, of course, they're not the only ones. And then, of course, there's the analysis of what comes out of the instrument. When we're talking about sequencing, all 4 of those steps, you really have to care about what your platform is. And so, of course, there's not a single sequencing platform, and it's reasonable to ask why not and what actually matters. And I think, these are kind of the 4 attributes that I think are worth considering when you're thinking about different sequencing technologies. There's, of course, a throughput, which is, you can think about it just in pure numbers of bases. But you can also think about it in other ways, such as if you have a bunch of different cancer patients, how much can you multiplex and have, samples from various people or different samples from different tissues from one person, for example. And you can also, when you're thinking about throughput, aside from how much data is cranking out of the instrument, there's, of course, how much labor and sample preparation that needs to be done. And so I do think that's relevant to the throughput, of course, because if you can't actually prepare samples very quickly, then that will also become a bottleneck. There's a yield, not just the number of bases you're getting, but how many of them are actual useful reads, right? If you're sequencing a lot, but it's not useful, and that's not so great. And then we'll talk a bit about read length throughout this lecture, which turns out to be important. And as we're thinking about all this stuff, of course, there's the quality of what we're doing, which is kind of related to the idea of the useful reads, right? So as you get a base, how sure are we that that base is what's there, and how sure are we that we didn't skip over one or double count one, things like that. And then there's, of course, the economics of it, which I already kind of alluded to when talking about labor, but there's, we can talk about these things as, per run costs or per base costs, what the total cost of the equipment is, and the reagents, maintenance, all those kinds of things. And this isn't supposed to be an exhaustive list, but I think as you're thinking about, if you're in the beginning of working on a project and you're trying to decide what sequencing technology to use, especially because they're changing all the time, it's useful to kind of go through these checklists and make sure that you've thought through all of these different, characteristics. So, how do you decide? Well, of course, back in the early days, there wasn't much of an option. Everything was done one at a time. And kind of the classic version of this, for those of you who know about it, are the Maxim Gilbert sequencing experiments. This year, I cut the slides to go back over that. It's kind of really cool classic stuff. And I think as a nucleoside chemist, I love it. But I've kind of learned that it's probably not the most important thing to emphasize. But I do want to bring up Sanger sequencing, which some variant of which a lot of people who do biology are used to, because it tends to be, the foundation of the technology people use to look at sequences one at a time when you're cloning a plasmid, and verifying that what's in the plasmid is what it's supposed to be. And so I'm not going to talk much about that, because from a genomics perspective, that tends not to be super relevant. There are times where you might want to validate something using Sanger sequencing, and I certainly don't mean to diminish the importance of Sanger sequencing, but just to say it's not going to be the focus of today's lecture. Instead, I'm going to talk about the three technologies here, focusing especially on Illumina. And Illumina has taken a huge amount of market share, and I think I still am focusing on it now, even though the technology is now reasonably mature, in part because of the motivation I showed at the beginning of this, which is most of the time when I open up the papers that I'm reading, a lot of the kind of relevant biomedical literature is really focused on Illumina still. And that probably won't always be the case, but they certainly continue to have a really important role. And so I'll emphasize that just so that we can go deeper in one of these. But I also do want to mention that here at Yale and in general, the biomedical community, there are additional technologies that are catching on. And the main difference between them and Illumina is that Illumina tends to be focused more on shorter reads, whereas these other technologies, PacBio, as it's affectionately known, so from Pacific Biosciences, these instruments, as well as the Oxford Nanopore technology, which you'll see abbreviated in some of my future slides, ONT. These guys, they're the main thing that distinguishes them in terms of functional output is that they tend to provide much longer sequences than you get from Illumina. And Illumina is referred to as short read deep sequencing, whereas these guys are long read. And I'm not going to go through all the statistics here, but I just want to point out a couple key things, which is, of course, if you look at these are mostly the statistics that I showed you on a previous slide of how we assess these. And generally speaking, the main advantages of PacBio and Oxford Nanopore tend to be that you get these really long sequencing reads. And so that's really great. But then when you look at the read accuracy, you'll see while these numbers are reasonably high, they're nowhere near as high as the numbers for Illumina. And in Illumina, sequencing still tends to have the highest read accuracy, and that tends to favor its use in certain types of applications. And so this is all taken from a review that really focuses on long read sequencing for anyone who's interested in it. I'll use at least one other figure from that review in this talk too. So what's the goal here? We can go in deep in any technology, and you could argue that we're wasting our time because it's going to become obsolete immediately. And yet I'm going to do that anyway with Illumina. And the reason that I want to do it is I really want to demystify sequencing technology. You guys are all critical thinkers, and it's worth thinking critically about the different steps and how to analyze the data you're getting. And while Illumina may or may not be relevant to your future, the point is that feeling comfortable going through and really thinking carefully about both the wet lab and the dry lab is relevant to to everyone's work when they're when they're using sequencing, and hopefully that'll come through. Oops, sorry. I think this. There we go. We're cleaner now, yeah. Okay. Okay, so how do you actually do a sequencing experiment? Let's start to think about this. And I want to start out by acknowledging, I don't know, has anyone used the YCGA? For those of you with your cameras off, you can
A couple of times.
hands. Okay, nice. So the YCGA is one of the cores on campus, and it has a ton of the sequencing facilities. It's certainly not the only place on campus that you can sequence, but it's the biggest operation we have here at Yale to do sequencing. And so I think it was Raghav who said it. Did you submit samples, or did you make your own sequencing library? Sorry, did you submit samples and have them make the libraries, or did you make it?
We directly submitted the samples to them, and they did everything for us, even right to the process where they gave us the differential expression file. So it was really easy for me to do everything else that I needed to.
Perfect. Okay, I'm really glad. So this is the point of this slide, which is you can outsource essentially all of this. And that's fine. But once again, the point of this lecture is to understand that when you outsource it, the people you outsource to are making choices. And you should at least be aware what those choices are and to be able to think critically about them. That doesn't mean that, and so if you look, this is once again a screenshot from a day ago of, the YCGA site so that you can see the prices of these types of experiments in case you're curious, but you can also see they have different types of library preps that they'll do for you. And as Raghav just said, you can go all the way through to analysis with them. And so that's great, but it is worth going through these things, no matter whether or not they're doing it or whether you're doing it. Thinking critically about these steps helps. And so, there's a sample preparation, of how you're doing the isolation library construction. There's how the sequencing works and the data analysis. And so each of those we'll go through in just slightly greater detail in a moment here. Before we do, and just partly for fun, I want to launch a poll here. And the goal of this poll is just to ask a question, just to kind of get you guys thinking about Illumina sequencing. So my question for you is, what is the most raw form of data that gets recorded in a Illumina sequencing experiment? And if you don't know, just guess, that's fine. I suspect some of you have more experience with this than others. All right, that's enough. If you haven't voted, that's okay. So I'm going to share these results. Can you guys can see the results? So it seems like people are pretty split about what it is, which is good. So I'm going to come back to this, hold this thought, and let's go ahead and talk about what actually happens in an Illumina sequencing experiment. Let's see. So what is the output? Well, the output, so meaning eventually what Raghav gets, although I guess if you go all the way to differential expression, you skip over this step. But, I think most people think about what they get from the YCGA as being a set of files, and those files are called FASTQ files. And a FASTQ file is something you can open in a text editor, right? This is your, the data that you tend to receive from the YCGA or your equivalent source. And these are also the data that are required for upload in most journals these days. I showed you in the Geo database in that example at the beginning of this. And these are called FASTQ files, and I'm just showing you a FASTQ format. And so there's a line of gobbledygook. There's a line that we can kind of infer what it likely means because it has A, T, C's, and G's. There is this plus sign, and then there is this other thing that looks like it's gobbledygook, right? And so, when you can look at this, it's like, okay, we kind of get it, right? We're asking for sequencing data back, and we're getting sequencing data here. And then we can go through and we can count this and be like, it seems like this is, probably, our sequencing output. So I think just from first principles, you can kind of squint at this and kind of imagine what must be in here, at least from the perspective of the sequencing data. But what's this, all this other stuff, and why do we bother even keeping it? So, and of course, I just showed you one line of a FASTQ file, but the FASTQ files that you get are usually on the order of, tens to hundreds to even billions of reads per sequencing lane. And these days you can multiplex your sequencing lanes. So, whether or not that's more than one sample is kind of an aside, but the big point is this is actually the data you get. And so I hope that over the next few slides, I will be able to demystify what these guys are, how these data get there, so that when you get your next FAST2 file, this is a nice familiar file without the gobbledygook, but rather with something that you can actually interpret. Okay, so first of all, how long are the reads? The reads themselves, so each one of those lines is a read, and the reads can have different lengths. This particular file that I was showing you has a 75 nucleotide read there. It turns out that that depends on how you run the instrument, and we'll talk a little bit more about that. But I just want to say, remember earlier on, we were talking about Illumina technology, and we're saying they're short reads. So what does short mean? Well, short means between 50 and about 250 nucleotides. Some rare cases you can go up to 500, but this is kind of the standard for Illumina technology. All right, so where do they come from? So let's just go through in kind of some detail here and kind of seeing where people are with molecular biology. I've kind of learned to simplify this a bit. I'm going to have one extra slide to go into greater depth on the molecular biology, but let's start with just a basic idea, which is you get DNA, right? So maybe you get DNA from a patient. It sounds like a couple of you guys had done cancer work. If you're trying to do bacterial genomes to try to predict the RNAs there, in order to get RNA secondary structure predictions, of course, you need someone somewhere got a bacterial sample and sequenced it. So you actually have somewhere the actual DNA itself, right? And you can extract the DNA. How many of you actually, or at least those of you who have your video on, did you guys do a lab at some point where you spooled DNA out of a sample from a strawberry or something like that when you were before? Okay, I see some people. So you guys know, like, you can get DNA, right? This is a thing. There are kits that let you get it from various samples, and we can talk more about that, the different sources if you want. But basic idea is you get genomic DNA. But and we're focusing on genomes right now, I'll mention again on Monday, we can go back to other applications of sequencing. And the basic idea here is that the DNA itself, we're going to need to make short reads of it, and we're going to get lots of them, right? And so in order to do that, we're going to start out by fragmenting the DNA up into little pieces, and that serves a few purposes. One is, I think it's kind of intuitive that if you're doing shorter reads, you might want shorter pieces of DNA. There's kind of some gut appreciation. There's also a handling point here, which is that giant pieces of DNA are difficult to work with biochemically, and so it's a lot easier when it's sheared up. And so there's a lot of reasons to shear up the DNA in this fragmentation step. But once you have that DNA, at least in Illumina technology, but in most technologies right now, it's not really the case that you can just take DNA and throw it at an instrument and have the instrument tell you what the sequence of that DNA is. People are working towards that. That's certainly a goal in the field, and you can say that some people are coming closer to that, but you need some way so that DNA can get read by the instrument. So we're still in the world of biology and chemistry here. And so we need some sort of way to adapt your DNA into a format where you can see it. And the answer is that you can put on sequences that are known onto the end of each DNA fragment. And so the concept here is right now we have no idea what any piece of our DNA has and what A's, T's, C's, and G's it has. But if we do some molecular biology tricks, we can add on adapters to the end of our fragment. So then, even though we don't know what's in the middle, we at least know what's on each end of our fragment. And as you'll see, that's going to become really important in the way that the sequencer actually works. And so once you put these on, you can make and now once you have information about what's at the end, this is sufficient. You don't necessarily need to amplify it, but frequently, if you don't have a lot of material, then it's possible, once you know what's on the ends, is you can use techniques like PCR to then amplify this without having to know what's in the middle. Since you know what's on the ends, you can make primers and you can amplify this material. So this is kind of nice, and then you can get your library, and there's all sorts of quality control, and we can talk about that more if people have questions about it, especially if we have time towards the end of this. But what do we do with it? Okay, so now at least we've converted raw, totally unknown DNA to smaller fragments that we would be able to put on a sequencer. The next thing we need to do is we need to get these things into the sequencer in a format that works. And it turns out that Illumina uses something that is important, which is that it can be really difficult to sequence individual molecules of DNA. And as we'll talk about in a little bit, there are other technologies that can take individual molecules and look at them. But in general, there are a lot of challenges to looking at individual molecules. And so we have a huge library, right, of different DNA fragments with different sequences. And if we just amplified our library, the problem is they would be all mixed together. And so the trick that Illumina uses is they have what's called a flow cell, which is essentially the equivalent of like a glass slide, more or less, that has the equivalent of PCR primers that are covalently attached, meaning they're physically attached to the slide. And since we know the ends here, we can design the primers so that they'll hybridize to our library. And since every member of our sequencing library has the same ends on them, all of our members of the library will end up hybridizing somewhere on this flow cell. And the cool thing then is that you can actually do PCR right on the flow cell. And because each one of these guys has two ends that we know and they're different, what that means is you can do PCR, but instead of having the primers be free in solution, the idea is the primers are all covalently attached. So you'll only be able to amplify locally, meaning you'll make a whole bunch of copies of this DNA, but only when it reaches over to a nearby primer that's immobilized on the glass surface. And so the cool thing that does is it makes these clusters, and the clusters are all copies of the same original molecule that hybridize to any given spot. So the trick with Illumina sequencing is you have this sparse hybridization, and then you do this PCR where everything is immobilized, but you get these little spots of copies of each one of these molecules. Why you're going to do that, we're going to get to once we get to the sequencing. Does anyone have any questions about how the cluster amplification works though? This is one of those steps that I recognize that depending on your comfort with molecular biology, the words I said may be more or less easy to follow.
Can you repeat that once again? Because I kind of lost you, specifically at the step where you said it hybridizes with the sort of the adapters that are on the flow cell itself, right?
Yep.
And then it amplifies them, but how does it really sort of cluster? I couldn't really understand that.
Right. Yeah, so that's a good point. I'm realizing, so I cut out some slides that I thought were redundant here, and I kind of wish I didn't. So let me see if I can use the annotation tool here to help. So the idea here, and I apologize, I'm using my mouse. I don't have a proper stylus set up right now, so apologies for this. But so imagine right here. So can you guys see my annotation? So not good. Okay, so this guy right here is the one that's actually covalently attached to the slide. And then the idea is that through a hybridization reaction, one of our library members has base paired on. So through base pairing, we've captured one of our many library members. And once again, we don't know any of the sequence in the boxed area, but we do know the ends because we added those adapters on. And the idea is once you have one of these guys on, there's a bunch of other primers on the surface of this flow cell nearby. And so if you do the equivalent of a PCR reaction, what can happen is that this DNA sequence that's right here can lean over and really bend over and find one of the other primers that's there. And then this primer can be extended in a PCR reaction. Kind of, yeah, I think you guys can get that. So it can get extended, but the cool thing is it will extend all the way through. So then the next step of this is we went from having one copy of this guy to now we have two copies, right? And then of course, these two copies through future steps of the PCR can become 4 and 16, et cetera, or 4, 8, 16, yeah, you get the point. And eventually make these clusters of hundreds of copies of the same sequence. And the point is that there's another DNA that's over here, and it's going to copy, but it's only going to get copied into regions that it can lean over And these guys each lean over, but they all stay very local. So you end up making local copies on the flow cell. Does that help, Raghav, and anyone else? I suspect you're not the only one who had questions about this.
No, this makes absolute sense. Thank you so much, Matt.
Great. Any other questions about this? Oh, good. Only works if these, these DNAs are packed far enough apart. And of course, you could go to very dilute and only put one molecule per flow cell, but that'd be very wasteful. And then you wouldn't have to worry about overlap. If you make it really dense, of course, they would be too close and the clusters would start to overlap. And so in fact, the reason we have really highly skilled technicians at the YCGA is in part to try to make sure that you're always loading at maximal level to try to get as many clusters as you can without too much overlap. But actually, as you're going to see that plus mark in the FASTQ file, that's partly there because there's actually QC steps that are going to happen to make sure the clusters aren't overlapping. And if they do overlap, then they just get thrown out from the analysis. So it's a very good question. When you put adapters on, and once again, I'll have another slide on this, but when you put these guys on, these adapters, the idea is that we know those sequences. So we're using molecular biology to glue known bits on to the end of something. So imagine, for example, you took a book, you ripped it up, but then you took a new cover and put one of the pages in because you know the cover that you used. If you had tons of the same copy, we know the front cover and back cover, but we don't know what's inside. But at least now we know what the cover is. It's kind of similar. We know the very first nucleotides and the very end ones because we put them on using molecular biology, where we had known sequences that we added. I'm going to move on, but I want to make sure, especially Stella's question gets answered about how we actually get the sequences. So right now, all we do is we have clusters. And the reason we did that was to amplify. So let me get rid of these very messy drawings if I can. So now we have these clusters, but what good are those? And the answer is Illumina does something called sequencing by synthesis. So now we have a whole bunch of copies of the exact same, sequence. And then what we're going to do is, and we know the ends. So what we can do is we can take the known end and then add one nucleotide to it. And that one nucleotide could have yellow dye, green dye, blue dye, or red dye on it, depending on whether it's an A, T, C, or G. And then what we can do is we can pause. So we would add one base. And if we added all the bases together and we didn't do anything fancy, they would all get added by a polymerase. And so that wouldn't be very helpful. So what we do is we add one base at a time and we use chemistry that blocks any further addition. So we add exactly one base. And then now each cluster should have exactly one first base after the adapter. So kind of going with this, if we're sequencing up this way, the idea is that we're going to add the base that's right here. And depending on what that base is, it could be an A, T, C, or G. And if you imagine that it's a G, if we had a C floating around and that C I should have made blue, it has a fluorophore that's blue on it, then every single molecule in this cluster will all get a blue fluorophore on it. And what that means is then we can just take a picture after one cycle. And if we had a C at this position, this entire cluster will show up as a blue spot. And if instead in some other cluster, this was an A, then a T would go on, right? Because A hybridizes to T. And that ends up causing that spot to all have the color T, which in this cartoon is green. And so the idea is after one round, we can figure out what the first base is. And the cool thing is after you do that, you can just remove the protecting groups that are blocking the elongation of this in the fluorophore, and then we can add one more base. And so in my cartoon, I'm just going to imagine that's an A, and then another base. And each time we're going to add one base, take a picture, and then deprotect, do another base, add a picture, deprotect, add another base, and continue that cycle over and over again. And every single time we're going to take a picture, and each picture is going to tell us for that cluster what base was present at each location as we proceed through our molecule. Let's see. So I'm seeing some questions that are kind of higher end questions about paired end read. So we can come back to that. I want to make sure everyone gets the basics and then we can talk about fancier stuff in a moment. The one question that I really want to get at here is people are saying, is there going to be a bias when you do the sample preparation? And yes, that's exactly the point of this lecture, is that all of these things are real molecules, and it's really hard to get every piece of DNA to behave the same as every other one. And so when you're going through and doing these sequencing experiments, there are very real sources of bias at pretty much every single one of these steps. And we'll talk about it more in the molecular biology step in a moment here. That's the question before about how we infer the sequence from doing this. Do you now feel like I've answered your question about how we're actually getting the sequence?
Yeah, that makes a lot more sense. Thank you.
Good. Anyone else have basic questions about how we sequence by synthesis with fluorescent bases and take pictures at each step? Cool. All right, so I'm gonna keep moving, but once again, there are some higher end questions that we can come back to. And it looks like it cleared, which is what I was actually trying to do. Okay, so here's the more in-depth stuff. I intentionally call this slide optional so I don't scare people, because my experiences are sometimes physicists and other people in the audience who just really don't know their molecular biology, and that's fine. You don't need to know this, but because there are a bunch of people who do care about this level of detail, it's really cool. And so pretty much this gray thing, this is your DNA sequence. So you shear up the DNA. And what people really do is they go through, and this is one, there are multiple ways to prep DNA. So I'm just showing you one option. But you can do what's called end polishing, which means essentially taking a polymerase and filling in everything to make sure that there aren't gaps in this so that it's really double-stranded. And then the thing is that it's actually really hard to ligate onto a blunt end, meaning if you have perfectly paired, complete double-stranded, it's actually a little bit hard to ligate onto that, as some of you may know. And so there's a trick where you can use a polymerase that it just adds an A overhang on the end. And so now we have just a little bit of a sticky end, which makes it so that then we can take our adapter and ligate it. And I bet a lot of you have done ligations before, and your adapters, so if you just have a T that'll pair with the A, you can get this to go. And I didn't talk about it, but you need to make sure your phosphates are in the right place, which those of you who've done a bunch of molecular biology will know, this is one of the important steps of making sure ligations work. But there's something really funny about this. It's really cool, which is that you use these Y-shaped adapters, and that's not normal in molecular biology. Usually you just ligate two pieces of linear DNA together and stick them together with a sticky end. But these Y-shaped adapters, and once again, I won't have time to go through all the details of it, but those of you who are asking about the paired end sequencing, the key to this is what we'd like to do is we'd like to desymmetrize our molecule. So what's on the 5' end of it and what's on the 3' end of it look different. Sorry, I thought I had my notifications off. Let me just pause and take care of that. Sorry for that. Oh, I do have do not disturb on. I'm not sure why that. Oh, it's because of my iPad screen. Sorry. Anyway, so the idea here is that if you think about what would happen if you just added a linear molecule of DNA on, then the 3' end would just be the reverse complement of the 5' end. And once again, I know I'm talking over some of your heads. Don't worry about it if I am, but I think it's cool. And for those of you who are into these details, it's pretty neat. The idea is if you use these Y-shaped adapters, what you can see is that on this side, we have this P5 adapter. But if you think about what goes on the other side, well, the answer is the other side looks like this, which is that it has the P7 adapter. And in fact, if you look at the product of this, you'll see this is the same Y adapter, it's just flipped over. But the net effect of using this Y adapter where this part is not complementary to this is that now we have different sequences on the 3' and 5' ends of your DNA, which makes it so you can really add independent barcodes and independent sequencing adapters in order to do this. But so the net effect, just to zoom back out, regardless of the Y-shaped thing, which if I lost you on that, that's okay. But the point is that by doing these steps, first of all, we get this on, we get to go through a high yield ligation reaction, and we end up with this DNA insert where we now have the P5 and P7. And I actually call them that, even though those are obscure jargony names, because these are actually the adapters that you find on the Illumina flow cells. And so what I've just shown you here is kind of the cartoon scheme of how the molecular biology works so that you can add the same adapter. So you have exactly one adapter, even though you have a very complex library of lots of different fragments of DNA. And even though you have tons of different DNA inserts, we now know for any given sample exactly what's on the 5' and 3' ends. And just to go, once again, a little bit deeper for those of you who care about the details, these blue and orange parts, these are where the sequencing primers are so that the sequencing can start right on your sequence of interest. And the one other point I want to make is that you can add indices. So if you imagine that you have 96 different samples in a 96-well plate, you can use adapters that have 96 different known indices in each one of those wells. And the advantage of that is that then each one of those wells is going to end up with something that looks exactly like this, except that each one will have a different index. And what that means is we can take those 96 different samples, put them all together into a single tube, let them mix together. And when we sequence, as long as we sequence across this index, we can then figure out which of the 96 wells our sequence read corresponded to. And so this index is a secret to being able to multiplex. And in fact, then you can put indices on both sides of a paired end read. Okay, so I'm gonna finish this. Once again, I wanna recognize this is optional. I know this went over some of your heads. I also hope that those of you who are doing some of this stuff just added some of the really cool texture for you. Are there any kind of general questions about this? I see some thumbs up. That's good. Thank you.
Matt,
Yes.
I was wondering, why is the clustering, sorry if this is jumping ahead, but why is the clustering necessary? Why not just have separate flow cells or wells or?
Yeah, so, okay, so the question about why is the clustering necessary? So some of you may know it is possible to pick up individual fluorophores using single molecule techniques. That is certainly possible, and you'll see that their PacBio, in fact, uses single molecule sequencing. It turns out that's challenging, though, both because of the optics and things like that, as well as you have to worry about photo bleaching. And so there's a bunch of logistics associated with single molecule techniques, which is harder. So the pre-amplification is one relatively easy way to make it easier to do this sequencing. But no, it is not strictly necessary. And we'll get to that. But the bottom line is that you're absolutely right. In principle, it's not in practice. There's a reason that this was a scalable technology that worked well. And I think that's about as far as I'm going to say.
Thank you.
Great. Let's see, just doing a quick thing. Yeah, sorry, the SPRI stuff. So for Ivy's point, yeah, so the SPRI stuff, for people who know it, it's essentially a really efficient way to do size selection for different stuff to get rid of excess adapters and things like that. And that's a level of detail I just don't have time to go into here. And is the insights the same as the barcode? Yes, those tend to be used interchangeably, so yes. Let's see. Okay, so I'm going to keep moving, but I do want to bring up the point that was raised earlier, which is, yeah, there's all sorts of potential source of biases here. And this is why I care that you guys know how this works is, you know, there's selective PCR amplification. It's known that really G-rich regions frequently can get lost in library prep, in part because it's hard to PCR amplify, things like that. If your shearing doesn't go smoothly, you can imagine really large fragments. When they do this PCR reaction on the cluster, they can make spots that are too big and are more likely to get thrown out. And enzymes do have specificities, and you can, lose some sequences because of it. So there's all sorts of reasons that this does this. Of course, there's a huge industry here where people have really optimized these steps. So, by and large, these aren't huge problems, but you do see them popping up from time to time. And it is good to keep in mind. Let's see, and one thing I'll just mention is mostly what I'm talking about is doing bulk DNA-seq. The library preparations here that I'm talking about. This tends to be if you have lots of sample. Many of you know that there's actually single cell versions of DNA-seq and things, and they tend to use different types of library preparations than what I'm showing here, in part because this type of molecular biology is hard to do on a very small scale. All right, so let me just come back and see if we've learned anything here. I'm going to relaunch the poll and say, and I will acknowledge there's some ambiguity in the way I ask the question. So if you realize that, that means you're paying attention. But all right, that's enough. I think we can go ahead. But yeah, so right. So the idea here, right, is that the majority of the so the way Illumina actually works is it records the images. And if you want to be technical, I realized only after writing this poll that depending on how the photo multiplier tube in the, when collecting the image works, you could argue that maybe we're looking at current. So those of you who said D, if that's your reasoning, that's fine. But yeah, mostly the idea, right, is every single image, every single cycle of sequencing, you get an image with different fluorophores locations there. And then, of course, then that gets processed, and that process leads to an interpretation for each spot as a string of letters. So now let's come back to what you actually get out of paired end Illumina read here. And this is what I'm hoping will be satisfying here. So what is all this gobbledygook? Well, this first line here, what is it? Well, it turns out this is all the information you need to know about the actual sequencing experiment that worked. We started with the instrument. So this HWIST, this is the name of the instrument. There's the flow cell, right? Because you can imagine you have multiple flow cells. So this is just, kind of keeping track in case a flow cell was bad or an instrument was bad or if something was weird. That's nice. But then you have a read ID. So that's going to be the unique read ID of the, for any given within that flow cell on that instrument, you're going to have some number for it. And then the cool thing here, D, you have the coordinates. What do I mean by coordinates? Well, what I mean by coordinates is that this is actually, right, this is in an image. Right, so when you're going through the images, you have an X and a Y axis, and I'm not going to go into the details of how they do this, in part because the different Illumina does it slightly differently. But the basic idea, right, is any given spot is going to be in the same location in all those images, and that's how we're figuring out what spot is what. And therefore, we can use the coordinates of that spot in order to figure out, which spot we're talking about. And in principle, you could go back to the images and find the spot that corresponds to this read, right, and actually look at it. And then we have which read from a paired end sample. So this is the complex stuff that I'm not talking about, but you can actually read forward and then reverse on the exact same cluster using two different primers. And in fact, you can do multiple primers if you want to get indices and barcodes and stuff. And so this just tells us which read it is. So in this case, it's 1, in this case, it's 2. So these are the two paired end reads from otherwise the same instrument and location. Right, you see everything else was the same. It's just read 1 and read 2. And then this last bit, this is which index from a multiplexed read. So this is a 6 nucleotide code that told us where in our, in our imaginary 96 well plate we pulled the sample from. Does that make sense to everyone? So, all right, that's pretty useful. Then this guy, well, here's our machine's interpretation of every single one of those photographs, whether it's A, T, C, or G there. So that's pretty nice. That's the sequence, and we know how it got it now. Now this plus here, and if you guys get your sequencing from the YCGA, every read will have a plus there. But you can infer the reason there's a plus there is because sometimes there's not a plus there. Those are the ones that are minus, and this is a quality score identifier. And essentially this is to say that the YCGA takes all the reads that suck and they get rid of them before they send them to you. And so all the ones that didn't have a plus don't make it to you. So hopefully all of your ones have a plus there. And then, and so the reasons you might not, well, you can imagine if you have overlapping, spots or spots that don't show sequence that you expect, and you can imagine all sorts of other quality control metrics that they can use. And I actually don't know what the most common reasons that they get filtered are. I suspect it's overlapping spots, but I'm not sure. And then finally, there's something called the quality score. So why would you want a quality score? What's that about? Any guesses? Someone has to. Essentially, right. And the idea, right, is that we're interpreting photographs of our, fluorescent images. And sometimes we may not be quite sure what that spot was, either because, some are a little bit dim And it turns out that over time, each cycle, you can sometimes get better or worse images, and it tends to get worse throughout the run. And at some point, it might get bad enough that you don't trust it. And so instead of the machine telling you, okay, I'm going to just make an arbitrary call to stop trusting this, we're just going to have a whole bunch of metrics about how bright it is and how sure we are it is a T and whether the shape looks right and all those kind of things. And then we're going to compile that information into a quality score. And then people can use that quality score when they're doing things like alignments to figure out whether they care if there's a mismatch. And of course, we might care less if there's a mismatch if the machine wasn't sure whether it was a T anyway, than in a case where we were quite confident that our read had a T there. So that's the kind of thing that's there. So this is really the main purpose of this lecture to me. On the other hand, I do want to cover some other stuff. And so I'm going to speed up a little bit for the rest of this. But I just want to emphasize really what I'm hoping you got up to this point is that sequencing is a real thing. It's real molecules. We make it in. You can understand the molecular biology of how it gets there. You can understand what the instruments are doing in the raw data. You get out reports on those things, and is worth taking seriously and looking at and making sure you understand what's in it. And in the case of Illumina, anyone who's worked with a FASTQ file now should be able to open up and know what they're looking at. And if that's all true, then I feel like I've succeeded. And I'll just try to go relatively quickly through some other things that I think are important to cover between now and Monday. Does that sound good? Cool. All right. So why do you end up getting lower and lower quality? Well, you can imagine in your cluster, no reaction is perfect. And so each base that you add, if it's not 100% added, or if, for example, something happens that hurts the chain or anything like that, you can end up having one that gets off by one cycle. And if that happens, then while almost all the molecules have a G going on, another one might have a C going on. And eventually, if you get off by enough, you get out of phase, and eventually you'll get a statistical mixture of a whole bunch of different bases, and you'll be less and less confident of what you're looking at. And so pretty much part of what limits the length of Illumina reads, and part of the reason the quality gets worse and worse, is because you can get out of phase as you're going through the sequencing. And that's what people have been optimizing in order to make longer and longer reads. The other thing, of course, is that in order for that PCR to work, the length of the inserts actually matter. And so both of those matter for doing this type of clustering and for getting the reads to work. So, you go through and you get billions of these things in principle. And then, of course, the next question becomes, how do you take all this information and turn it into something useful? And we'll continue this a lot on Monday. But let me just kind of go through and say, first and foremost, most of the time for a lot of you, you're going to be sequencing in organisms where we already know essentially what it should look like. And the net effect of it is that you can just take a reference genome and do an alignment to actually say, where in my reference genome do I find this sequence? And so I'm not going to have time for this just because we spent some time. I'm really happy we discussed stuff earlier, but I'm just going to point out that, of course, a mismatch between your data and the reference genome could be because there's an error introduced during the molecular biology. It could be because there's an error in base calling, and all of these could be the case. The correct answer for this is D. Or it could be that, in fact, your reference genome just doesn't, there's a single nucleotide polymorphism, for example, in the individual you're sequencing that's different from your reference genome. And so in all of these cases, you would have, a read that's not a perfect alignment to your reference, but nonetheless, in most cases, because you're, using references where you have high knowledge about the sequences, you can use the reference genomes to learn a lot about where all your sequencing reads go and what's different between your experimental sample and your reference. Of course, there's some bookkeeping to do with all this. Is just like trying to get a sense, okay, so we have billions of reads and there's 75 nucleotides, and like how many reads do we need to think about the human genome? And so why don't we go through and just look at a bunch of different genomes for a moment and think about how big they are. And just, I want to remind you, so a kilobase is 1,000 base pairs, right? And a megabase, right, is 10 to the 6th, and a gigabase, 10 to the 9th, and terabyte, and so anyway, so on. And I think you guys know your metric, hopefully, so this should be a surprise. But the point is then we can look and we can go through and kind of benchmark on the human haploid genome, haploid meaning one half copy, right, because we have 2 copies of everything. is about 3 gigabases of information. And so you can just go through and you can see some genomes are much smaller than others. My lab, for example, uses fly cells a lot when we're developing technologies because they're about 20-fold smaller, which means you get about 20-fold more bang for your buck when you're doing sequencing in a complex eukaryote. And so then we apply things to humans, but we work it out in flies first. You know, and so these things are there. And this just gives you a sense. But like the question is, does it work out? So if we have on the order of millions of reads that are 75 nucleotides long, you know, does that give us enough bases to cover a human genome? And the answer is, yeah, it does. It works out about right. You can play with it. And the reason I'm not being more precise in this is how much coverage you mean, like how many times you have to read each base becomes important depending on what application you have. So if you care about every single base for sure and how many copies of it there are in your sample, you need a much higher coverage. Whereas if you're just kind of getting a once over of these things, it becomes less important. So the point is that we're in the right scale, which shouldn't surprise you because of course this is the whole point of these deep sequencing. They kept getting optimized till we got to a point where we were on the right scale. The right scale for the human genome, you know, the victory was declared in 2003. I'm saying declared because, of course, there's still parts of the human genome we're not positive about because of repetitive regions, areas near the centromere, areas near the telomere, but it's gotten really good now. But, you know, just there's some cool numbers, but in the interest of time, I'm not going to go through them, just to give you a sense of how much technology has improved over the last, you know, couple decades. Oopsie. So, you know, once you have all these reads, it's worth mentioning that, of course, if you have tons of little reads, that's a little bit of a pain in the butt to try to figure out what a long genome looks like from little reads. And in principle, you just get over all their overlaps, right? And if you have all their overlaps, eventually you can assemble an entire genome. In practice, it's not quite so easy. You can find a lot of overlaps with your short reads, and then you can make long stretches of them, but frequently you get limited either because of repetitive sequences or a lot of other regions. So you can make these things that are called contigs where you can kind of assemble them, but you still have to figure out how all your contigs actually fit together into bigger and bigger pieces. And once again, I don't really, because of time, have time to go into all the details of the scaffolds, but the concept is you have to get all of the overlaps to come together. And thankfully now these long reads that I promised you I would get back to, this is where the cool new long read sequencing, either the PacBio or Oxford Nanopore, really allow you to assemble, you know, data across much longer length scales. And in the interest of time, I'm just going to say that PacBio uses single molecule data, where nucleotides is still fluorescent based. And the idea is you have the nucleotides going in and out of a polymerase, and their dwell time tells you what base is there in the complement strand. And so you can read this review if you want more information. Whereas Oxford Nanopore has this amazing thing, which is that as DNA goes through, it actually changes the electric current that's going through and really the resistance across these nanopores. And so you can use a squiggle of how that perturbation happens to figure out what the sequence is of the DNA that's going through the pore. So that's both very cool technologies that are growing. And what you can see is if you have the mean size of those things I call contigs, right, which are like this stuff we can just get through overlapping versus the number of contigs necessary to get the genome, what you can see is that the Human Genome Project required tons of contigs because the average size was very small. So you had to seed a ton of them together. Whereas now with these new techniques, you get these really long contigs that are on the order of megabases. So now you have many fewer of them to put together, and you can start to get a lot more information about, you know, how the chunks of the genome come together. And especially this is important in repetitive regions and low complexity regions that if you think about for a little bit, you'll realize are really hard to get when you have short reads. So let's see. Most of this is going to be wrap up. So I'm just going to kind of foreshadow what we're doing on Monday and then wrap up here, which is just to say, you know, kind of course, getting all your A's, T's, C's, and G's is only part of genomics, right? You can, okay, so we can get a genome, but a genome, right, like this, I give this to you and ask you about biology, you know, this doesn't tell you anything. right? Like you need some way to read this, some way to annotate it, some way to know what you're looking at. And of course we know the genetic code, but that alone only gets you so far. We need a lot more. So we need to know where in this are genes, and even the genes themselves, there's a lot of nuance, like what different isoforms are there? Are they expressed or not? You know, how are they different between individuals? Are those things conserved across species or not? Or, you know, how are those genes that do encode for proteins or non-coding RNAs, how are they regulated? And so there's all sorts of sequences that are regulatory. And of course, even more than that, A's, T's, C's, and G's aren't the only important hereditary information. There's also something that's on top of that. Epi means on top of, and epigenetics is a study of all the things such as DNA methylation and histone modifications that happen on top of the DNA sequence that also are important for annotating the genome. So I'm not going to go through this just in the interest of time. We'll talk more about this on Monday, but the main points here are that we're going to talk about how you can use sequencing technology to go through and learn about all of these other layers of regulation of the genome. And it's kind of a fun back and forth between us using bioinformatics to be able to study how biology works, but also it's really how biology is annotating its own genome. And so we can learn a lot both about the biology and how we can study the biology by studying genomics. And so with that, I'm just going to wrap up by kind of showing you this slide. I haven't decided completely what I'm going to include as applications on Monday. So if anyone wants to email me, you can look over this, and if you guys are doing any sequencing applications that you think are fun, I'll see if I can kind of weave it into the arc of Monday's lecture. But otherwise, I'll wrap up here with just coming back to what I hope the take-homes were today, which is that sequencing technology is really central to understanding the genomes, and as you'll see on Monday, really understand a huge amount of biology. And because of the power of these techniques to really look broadly, the increase in throughput especially has made these data, you know, your figure 1A of a huge number of papers. And so, you know, as you go forward in the biomedical sciences, a sophisticated use of this data requires understanding both the biology as well as the sample preparation and the downstream computational analyses. And I think if you got those 3 points from today's lecture, then we're in good shape for continuing all of this on Monday. And please do come prepared with questions, or you can email me in advance if you had questions about today's lecture that I didn't get a time to ask. Normally I would have tried to save some time for a Q&A. So thank you everyone, and look forward to continuing this Monday.